Welcome to the Research Vessel Atlantis. For the last week, this boat has been the home to 24 scientists, 36 crew members, and we've been studying the methane seeps at Hydrate Ridge. Now, we're coming into port in San Francisco. And before we get off the boat, I'm going to take you on a quick tour of Atlantis.
That looks good. I'm going to go ahead and move it.
Good deal. Awesome, looking good.
So we're here in San Francisco, made it into port. Here with Baxter, a Jason engineering assistant. So what's going on? What are the components of Jason?
Well, this is basically the front end of Jason. We have the two manettes for actually doing things underwater. We did a lot of that for collecting and deploying and taking samples. What I'm standing on right here is the basket. It's basically where we put all the scientific gear. This will hold anything from your push cores to your sample markers to optical modems and all sorts of crazy equipment. We have a number of cameras all over Jason. This is the science camera. That's a huge HD camera that will give us movie quality images, and that you also take stills with that. We pilot with this camera primarily. Also these little micro cameras that will just get you a general idea of where you are, and then the raw camera.
So this time around, Jason was down for 137 hours. Yeah. Longest time ever. So how's it doing? Any fixes required?
As far as the biggest thing I think we were worried about was oil loss. After a while, you just get slight leaks, and because it's pumped, we're able to sustain that. But it lost almost nothing. It was phenomenal. So we really got the system locked down. A few little issues we're not related. We're not sure if they're related to the dive or not. It's just more maintenance stuff that we got those sorted out.
Cool. So when you're heading back out?
We'll be heading back out in a couple days.
Okay. So I'm sitting here with Greg Rouse now. He was one of the 4 main scientists on the mission. And he was looking at some of the macrofauna we pulled up, taking some pretty sweet pictures. So Greg, what were some of your favorite pictures?
Well, overall we got about 500 different records in this cruise and last cruise. But I had 3 real highlights, and one of the major animals that I work on that I'm interested in is this bizarre worm that's related to earthworms, but it manages to live on whale bones and even other bones we've now discovered. And so we put down some bones at 1,000 meters, and we got these worms after a year. They'd found these cow and whale bones, and they look like beautiful little flowers coming out, and they live on the bone with by really drilling into them like the roots of a plant, and then in these roots they have symbiotic bacteria, so they actually eat the bacteria, but somehow they're providing the bacteria with the elements of the bone like collagen or fat. And this is the first time we found them outside of California, and we found several of the species of California and got great photos of them, so I'm really excited about that.
What else you got?
I think the 2 coolest other creatures that we found was one was a deep sea nudibranch, and that's like a sea slug, or it's a mollusk. Like a snail, it's lost its shell. And one that we found is a little sea slug that eats hydroids, eats relatives of jellyfish, and jellyfish have, as probably all of people know, they get stung by jellyfish by these little stinging cells, and these hydroids that the nudibranch eats have a very vicious sting, but somehow the nudibranch can eat the hydroid, not get stung. It actually steals those little stinging cells and stores them in its little cells on its outside of its body and uses them for its own defense.
So normally nudibranchs are found in shallower waters? Is it rare that they're so deep?
The greatest diversity is shallow, yeah. It's unusual to find them deep, so it was really pretty cool to find them and to get beautiful specimens up and photograph them, so we're happy about that. And then the final thing we collected actually on the whole dive of this cruise was a beautiful hermit crab in a magnificent white shell, and it was sitting near an active bubbling methane site right next to the bubbles, and it was just sitting up on a mound and basically looking at us, not teasing us or taunting us, but we hadn't collected a hermit crab on either of the 2 cruises, and we've collected them down in Costa Rica. Okay. So I was interested if this might be the same species, so we got that.
These are the dual cold rooms on the ship. This is where we've been putting our samples. They're refrigerators, so they simulate the 4°C temperature at the bottom of the ocean. They're pretty much cleaned out by now, but you can sort of see some of the tanks and samples of water that we've collected. A couple days ago when we were finished collecting, these things were both full, which is a good sign. We've got one more stop on our tour, and that's the Rathburn Lab. Rathburn Lab studies foraminifera, and Ashley here will hopefully tell us what those are. So what are forams?
Well, forams are single-celled organisms that are about the size of a grain of sand, and some of them create a shell that is snail-like. They're very small, and they're some of the most abundant eukaryotic organisms of the deep sea, which is the largest habitat on Earth.
So you guys were here with us last year as well.
We were.
And what did you do then? You put down some experiments.
We put down some experiments, and we looked at them, and we actually found those epibenthic foraminifera, which means they live at the surface of the sediment water interface, or above it, have colonized the sides of these experiments. So here's, this is the frame of one of the experiments that we sent down, and it was covered in mesh along the side, and we found some pretty interesting species along the side, including Psittacipes dorfi, which is pretty important for geochemical work on their calcitic tests.
So you're studying like this modern environment to see how...
Yep, we do modern analogs. So we study modern environments to see what kind of signatures come from this modern environment, and you can apply that to the paleo record and assess how the environment has changed.
Okay, awesome.
Sweet.
Keep up the good work.
Thanks.
So we're taking our samples. We've got a lot of good stuff. And now it's a tour of the Atlantis, so thanks for stopping by.